Good morning, biology. Today we're going to be talking about the cell membrane and transport. Now, the basic, just general purpose of the cell membrane is to separate the inside of the cell from the outside of the cell. And again, cells exist in three-dimensional space, so they're going to be more like a sphere than they are, just a circle. The cell membrane is not as simple as just being made of phospholipids. It is the phospholipid bilayer, but there's also proteins in there. There's glycoproteins, sugars, cholesterol, proteins that have pores in them for moving molecules back and forth. It's a very much a complex structure. And the term they use to describe it is the fluid mosaic model. And fluid meaning free to flow. It's able to move. And then mosaic means a puzzle. So not only is it complex, it's like a puzzle with all these different parts. It's fluid because it's able to flow, but even the puzzle pieces themselves are free to flow in the membrane. So the transport proteins, cholesterol, it can all move and bounce around into different spots. And again, the basic purpose is to separate the inside of the cell from the outside of the cell. If you don't have a cell membrane, then you don't really have a cell, because then the internal pieces would just float off into space, into whatever medium that those parts are in, and they wouldn't be able to work together for a common purpose, and you wouldn't have a cell, you wouldn't have life. It transports materials into and out of the cell, so it can regulate what comes in and what goes out. And then it can also receive and respond to signals and ligands. And again, it's called the fluid mosaic model. There's three general parts that we're going to focus on. It's the phospholipids, cholesterol, and proteins. And again, one of the things we talked about with proteins for their purpose as a macromolecule was to get things done. And you'll notice that is a key point here as well. So going to the phospholipids, this is the most abundant component of the cell membrane. They are everywhere. There are billions and billions of these things. And their basic structure is their head likes water, and their tails do not. Their tails are known as hydrophobic. Their heads are hydrophilic. So if they are near a water molecule, the head will want to come close to water, and since the tail is hydrophobic, it wants to get away from water. So if you put a lot of these together, they naturally form a bilayer, because if you have water on both sides, well, the heads of the phospholipids want to touch the water, so they'll back up into each other where only the heads are touching water, because the heads are hydrophilic. And since the tails are hydrophobic, they want to escape water, but they can't escape the molecule that they're in, so they have to just face away from the water. So you get all these tails that are hydrophobic with all these heads that are hydrophilic, forming this, what's called the phospholipid bilayer. Now it's called that because it's made of phospholipids, bilayer because it's two layers of phospholipids. And there's another image of what it might look like. Cholesterol, we think of cholesterol as a bad thing, but you actually do need it. It stabilizes the membrane. If you get too much, then it thickens the membrane and it makes it too rigid. You want it to be flexible, but you don't want it to be so flexible that it ruptures. So it strengthens the membrane. It also lowers the freezing point of the membrane so that when cells are exposed to freezing temperatures, they can last a little bit longer before they rupture. Certain proteins are embedded in the cell membrane. Those that penetrate into the tails of the phospholipids are called integral because they are getting inside. So all three of these would be integral membrane proteins. If they go all the way across, we call that transmembrane because it goes all the way through the membrane. And then if they're only attached on the outside, we call these peripheral membrane proteins, just like your peripheral vision is on the outside of your vision. So what are some functions of these transmembrane, integral, and peripheral proteins? Well, one of them, if they have a pore going through it, well, then it can be a passageway, so it can be used for transport. Sometimes they have an active site that allows them to interact with certain food molecules. So in this case, they're functioning as an enzyme. They're catalyzing some type of reaction, perhaps on the inside of the cell for digestion or whatever needs to take place with that, whatever that protein is designed to facilitate. Some of them could be designed to receive signals, so it would be for cell recognition. Some work as cell surface identity markers, so it helps identify that that is a cell of the human body, so the immune system won't destroy it. It can be a site of attachment for the cytoskeleton, and then it can be used to connect two cells together, forming what's called adhesion. And then, of course, we talked about receptors. So there's also a layer of this glycolipids and glycoproteins that make up what's called the glycocalyx. One of the functions of this is it lubricates the cell, so it's able to not be so dry, and the cells can work together more easily and rub up against each other with less friction. And it also provides a layer of protection, and it's often called the sugar coating of the cell. So then all of those things we talked about, you can see them throughout this fluid mosaic model of the cell membrane. And then there on the bottom, they're showing you what that glycocalyx looks like under an electron microscope. So it's a pretty thick layer. It's given a lot of lubrication and a lot of protection. So then what are these different parts? What are these different proteins? What could they be used for? So in your notes, take a second, pause the video, and see if you can guess what each of these images is depicting in terms of the function of proteins in the membrane. All right, let's see how we did. A, you see two cells connecting, adhesion. B, it looks like they're breaking down some type of molecules, so that could be an enzyme. C, they're moving things across the membrane, so it could be transport. D, it looks like the two cells are interacting in the sense that some type of molecule is being recognized. So D could be recognition. E, it looks like it's attaching to the extracellular matrix. So the ECM is a point of attachment here. And it's also the cytoskeleton on the inside. So it can be used for attachment. And then F, it's receiving a signal, and that signal is being propagated throughout the cell, receptor. So when we talk about transport, we talk about the membrane, we say the membrane is semi-permeable or it's selectively permeable. And what that means is that the membrane can select, now it's not making a conscious decision obviously, but it can regulate what can pass through the membrane. Some things can freely pass, excuse me, and they'll just move from high concentration to low concentration. We call that passive transport. This is always going to be moving down what we call a concentration gradient. And all this means is if we have a membrane and we have a lot of salt on one side and we don't have much salt on the other, well, salt's going to move from high to low concentration. And water is also going to move from where there's more water to where there's less. So water is going to naturally move to the other side. And we'll look at several examples of this. We call that passive because it doesn't require any energy. Nature wants to move these molecules to be in equilibrium, so they're naturally going to move in those directions. Active, on the other hand, requires energy. It's going to require ATP, which is going to come from the mitochondria through cellular respiration. These materials are going to be moved from low to high concentration. Nature doesn't like that, so it requires energy to make that happen. So there, we're going to say that that moves against the concentration gradient. So here's some examples. Passive transport, you've got a bunch of molecules on the left, high concentration, not many molecules on the right, low concentration. Which way does nature want to move these molecules and to make them in equilibrium or balanced? Well, they're going to move to the right until you end up with an equal amount on both sides. Example of this would be tea bag in water. The tea bag in the water, as it heats up, the tea in the bag wants to diffuse into the water where there's less of that substance until it's equal. When we use the word osmosis, we're talking just about the movement of water. So if you have a semi-permeable membrane that only lets water move through, well, then the solute molecules can't go anywhere because they can't fit through this semi-permeable membrane. So even though these molecules want to move to the left, they can't. So the only way for nature to be in balance is for the water to move to the right until there's an equal amount of water and solute. So literally that water is working against gravity. That's how bad nature wants to be in equilibrium with solute and the solution and the solvent. So we call that equilibrium when the concentration of the substance is equal throughout the space in the solution. And that's going to happen naturally. If you just put, for example, dye into a solution, it's going to naturally mix. You put salt, it's going to naturally dissolve until it's evenly dissolved. If you heat up one room in your house, the heat's going to flow into the next room until both rooms are the same temperature. That's just nature trying to reach what we call equilibrium. I put that slide on there twice. I don't know why I did that. So again, water can flow directly through the membrane, but it flows best through these channels called aquaporins. Still passive transport, but it's being facilitated by this transport protein, or this protein channel called an aquaporin. And then if you don't remember, you do need to make sure you're familiar with the terms solution, solute, and solvent. And again, solute is just what is dissolved in something. Solvent is what has absorbed the solute. So solvent's almost always going to be water, and solute is almost always going to be like salt or sugar in the examples that we use. So then there's all these different kinds of solutions that you can get based on how much solute you have, and it can also be in reference to what kind of cell you're putting in the solution. So a hypotonic, the way I remember this is hypo-hippo. Hippos love water, so a hypotonic solution has a lot of water. So if I put a cell in a solution that's hypotonic, well, there's way more water outside of the cell than inside the cell. So which way is water going to want to move? It's always going to want to go from high to low, so water is going to rush into the cell. So that can lead to lysis, because if all the water pours in, because there's so much more water outside the cell, because it's hypotonic, well, it will flood in and it could make the cell burst, and we call that lysis. So for example, an animal cell, if this happens with an animal cell, it could burst. In a plant cell, it could flood that central vacuole, and the pressure could become extremely great, which could lead to the cell being super firm. But because of the cell wall, it may not actually rupture. So a good example of hypotonic solution would be if you took wilted lettuce and you could put it in a bowl of water, and it would actually firm up because there's less water in the lettuce. A hypertonic solution, on the other hand, is the opposite. This is when you have a ton of salt or sugar on the outside of the cell. So if you put a cell in a hypertonic solution, well, there's more salt outside. The water's going to go from where there's more water to where there's less. So if there's more water on the inside of the cell, then it's going to want to flow out, and the cell is going to crenate or shrivel up, and that could also destroy the shell of the cell. For plant cells, the water can move out. The cell pulls away from cell wall, and they call that plasmolysis or plasmolysis. And we're going to try to do that with onion cells in the lab one day if we're able to. So a good example of a hypertonic solution would be if you pour salt on a slug. What happens? Well, now you've put the slug in a extremely hypertonic solution because there's way more salt outside its body. So it pulls all the water out of the slug, and it will kill the slug. And then isotonic just means you have the same amount on the inside and the outside. So the cell will just maintain its shape because water will move in and water will move out at the same rate. And that's something to think about all the time, is that water is always moving in and out. Like even in this situation, it's not just moving in. It's just moving in at a faster rate than it's moving out until the point that it could lyse. So with red blood cells, we can think of that as a good example of an isotonic solution. In our blood, there's about 0.9% salt. Hopefully, we're constantly regulating it, and we're trying to regulate our calcium and many other nutrients and hormones. And if we have the right amount in our plasma, as we do in our red blood cells, then the red blood cells will not lyse and they will not crenate. They will just be the ideal shape. And there's an image of some red blood cells put into a hypertonic, isotonic, and hypotonic solution, and 3 plant cells as well. So we'll talk about types of transport. One of them could be facilitated diffusion. So we think diffusion, well, that's high to low concentration. Facilitated means something had to help it move across the membrane, had to facilitate that transport. Still going from high to low, but a protein is used to allow the molecules, because maybe they're too large or charged, polar, you had to have some type of pore or passageway for that molecule to move across the membrane. So you can have carrier-mediated facilitated diffusion. This would mean it's very specific to a certain molecule. Or you could have channel-mediated facilitated diffusion that is general, if it's channel-mediated. So it's just the channel. So it's general, maybe more than one thing could pass through it. So filtration is when the movement of solutes and fluids from an area of high pressure move to an area of low pressure. And this can actually help filter out what's in the solution before it goes into the cells. So it naturally filters it from high to low pressure. And this is exactly what you have with like a HVAC system in your house. It's a difference in pressure. And you also have these fans working. Computer fans create a difference in pressure. There's a difference in pressure from root to shoot in plants. And this can all be used to filter. If we move a lot of stuff at once, we call that bulk transport. And if we're bringing things in, we call that endocytosis. And if we're bringing things out of the cell, bulk transport, but it's moving out, we call that exo or exit cytosis. So we have different names for the different types of bulk transport. We're talking about endocytosis. If the cell is eating, we call that phagocytosis. So it's bringing in solids. If it's bringing in liquids, we call that penocytosis, which means the cell is drinking. And if you need receptors to facilitate that form of endocytosis, we call that receptor-mediated endocytosis. So when we import a certain substance using receptor proteins, as we see here on the far right, and a good example of that is how we remove cholesterol from our blood. It's a receptor-mediated endocytosis, and some people have genetic disorders where these receptors are not able to interact with the cholesterol, and that creates a problem because they can't get the cholesterol out of their blood, so they have chronic high cholesterol, which leads to hypertension, high blood pressure. And again, there's a zoom in of that diagram. You see the receptors here that have to be interacted with by the molecule of interest in order to actually create a vesicle and move those substances into the cell. And then exocytosis is when we move things out of the cell, the export of material. This will involve the Golgi apparatus, where these proteins are packaged up and modified, and then they'll be sent in a vesicle outside of the cell. Example of this would be the release of saliva from salivary glands, or the release of insulin from the pancreas. So what are the active processes? Again, that means we need to use energy. So we only would need that if we're going from low to high concentration. Because if we're going high to low, that just happens naturally. But if we're going from low to high, we're going to have to use ATP. So some examples could be a proton pump, sodium-potassium pump, calcium pump. We'll see this in our muscles. We'll see this in our neurons. And then proton pump is going to be really important when we talk about forming ATP in cellular respiration. And that is it. So just being familiar with the different types of transport and what some of these enzymes can do and how the cell membrane is set up. Thank you.